Okay, so the idea that makes it tricky to think about is that in glycolysis, we are all about ATP. But in gluconeogenesis, we have plenty of ATP in the cell. Like gluconeogenesis gets activated because glycolysis is not. Glycolysis is not activated because there's plenty of energy in the cell, right? So we no longer care about making or using ATP. The point of gluconeogenesis is to make glucose, okay? So we don't really care about this second reaction, the coupling of ATP, right? If we were able to reverse the whole thing and make ATP while making glucose, that'd be pretty awesome, but we can't. Instead, the point is to make glucose. And to do so, to make it from glucose 6-phosphate, all we really have to do is reverse step 1. That creates glucose from glucose 6-phosphate in a spontaneous manner, right? Simply reversing step 1 and using water to hydrolyze glucose 6-phosphate is spontaneous. And that exact same idea applies to step 3, right? The phosphofructokinase reaction couples phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate. We know that overall reversing that would be non-spontaneous, but the same thing goes here. If we decouple ATP, forget about ATP, and simply hydrolyze that phosphate, that is spontaneous. Yeah. Well, so we have seen a couple places in glycolysis where we use phosphate, right? Like phosphate by itself. So in the GAP DH step, we require this ATP. Anytime we want to make ATP in the mitochondria, we use phosphate. So the phosphate all kind of gets recycled in some manner. But right now, it's present in the cytoplasm, and it might get used in glycolysis. It might be used as a buffer for the cell. There's lots of things that it could do. It could be used to make pyrophosphate. Okay, so reversing 2 of the steps of glycolysis for gluconeogenesis is simple, right? There are 10 steps in glycolysis. We need to reverse all 10 of them. 7 of them are easy. 7 of them are at equilibrium. So we don't have to try. They're at equilibrium. The concentration of substrates and products, or concentration of metabolites, will dictate which direction it goes. The irreversible steps were step 1, step 3, and step 10. And steps 1 and 3 are easy. Reverse them, done. No ATP, nothing involved, it's spontaneous. But as it turns out, the last one, reversing phosphoenolpyruvate, that's harder. The pyruvate kinase reaction is spontaneous because conversion of the enolpyruvate to pyruvate, that tautomerization, is highly exergonic, right? So the second step, not the ATP, ADP part of it, the second step is the highly exergonic big driving force. So we cannot simply reverse step 2 and make this possible. Reversing step 2 makes the reaction much less spontaneous. We have a massive endergonic free energy here, right? If we simply try to reverse it, it won't happen. So there has to be an alternate pathway, okay? And this is where 2 new steps jump in, and we'll kind of briefly talk about them, and then we'll pick up here on Thursday, okay? So the idea here is that we have phosphoenolpyruvate. Right, so we want to convert pyruvate to phosphoenolpyruvate. And converting PEP to pyruvate is spontaneous in glycolysis. We couple it to ATP formation. We know that's spontaneous in glycolysis, but we can't reverse it. Okay? For gluconeogenesis, we need to be able to reverse it to get PEP to push back through gluconeogenesis, to be able to reverse glycolysis. For this to happen, there's 2 things that happen. First, the pyruvate enters into the matrix of the mitochondria, okay? In the same way we're going to see next week, it enters into the matrix to be part of the TCA cycle, okay? There's an enzyme in the mitochondria called pyruvate carboxylase, which takes carbon dioxide, consumes some ATP energy, and it adds carbon dioxide onto C3 of pyruvate. This makes an important TCA cycle intermediate called oxaloacetate. Okay, so pyruvate carboxylase is an enzyme that consumes energy, uses carbon dioxide, and creates a 4-carbon chain from the 3-carbon pyruvate. We then have PEP carboxykinase, PEPCK, that will convert oxaloacetate back to pyruvate, back to PEP. So the idea here is we start with 3 carbons, we add on a 2nd carbon. And then removing the carbon dioxide along with hydrolysis of GTP, this is enough driving force to convert the 4-carbon oxaloacetate to the 3-carbon PEP. Okay, 2 steps. Go from 3 carbons to 4 carbons by using energy. Go from 4 carbons to 3 carbons by using energy. Okay, so we will explore both of these in more detail on Thursday. The chemistry that these use is really kind of cool. We start getting to think about this molecule biotin. So biotin is an important cofactor, and whenever we're dealing with carboxylations, biotin is very commonly involved. Okay, so we'll get to see biotin working, and then we'll get to see how this PEPCK works as well. All right, told you stuff gets cool. That's more awesome now, right?